velocities, and we're talking about multiple sclerosis, and just on it, nerve damage repair, just on it goes. Okay. But for today, the myelin changes the anatomy. Where the axons are of the neuron cell bodies, the myelin around those axons makes the tissue look different. Where there are predominantly dendritic endings and neuronal cell bodies and glial cells, the tissue looks dark. Where there are places where it's primarily axons, the tissue looks light. Your book calls this gray matter and white matter. So as I bring the sensory information into the spinal cord, it goes into an area on the dorsal side. We call these the horns of the cord. Is it dark or light? Am I looking at a different picture than y'all are looking at? Okay, so it comes in here and it goes into this dorsal horn. Y'all see that? Into a dark area, an area dominated by cell bodies. Now, as this arrow comes this way toward the brain, we're going to see, now this is a cartoon picture of it, so it's all kinds of colors here, and I apologize. You can see all the horn stuff here, the gray matter in the center. All this area is supposed to be white matter, but it's color-coded here for teaching purposes. And notice how it's color-coded here with little circular areas of the spinal cord. You see that? Now, when we get to the nervous system, you'll have to learn all of these. When a student leaves my anatomy class and they go to the hospital and they see an image of a spinal cord with something wrong with it, a student who's taken a good anatomy class should be able to look at that image, and if you know what part of the cord you're in, you should be able to say exactly what the deficit is. Is it a sensory deficit? Is it a motor deficit? And where? Okay, so this is the first round here. Once the sensory stuff comes into the dorsal side here, it makes a synapse, and then it turns and sends the information to the brain. Now that's information leaving the neurons. Is that on the dendrites or the axons? That's on the axons. And if the axons are myelinated, they are white. So all the areas around the horns of the cord are bundles of myelinated axons carrying information to the brain. And they're organized. They don't just go through in a random way. They're bundles, almost like little fiber optic cable networks, where particular kinds of information are found in these different bundles. And so this particular bundle, you can see this one, is called an ascending tract. And you can see that the tract is going to go from the spinal cord, finally terminating up here in the cortex of the cerebrum, passing through, again, information you can never forget, passing through a structure called the thalamus. This is one of the primary ways that pain, temperature, pressure information get to your brain. They pass from your skin into the white matter fiber cord tracts, so we call them tracts of the spinal cord, that are then delivering that information to your brain through the thalamus. These pathways are called the spinothalamic. The spinothalamic sensory pathways. They're arguably the most important. Now, I think if you really pin me down, I would say this stuff up here, the dorsal funiculus, is even more important, but we can talk about that later. Look at the picture here now. Where is this information? In your spinal cord, where is this touch and pain and temperature information?
Where is it coming from?
Where is it in the spinal cord? I know where it's coming from. It's coming from the skin, from the Pacinian corpuscle. And I know that it goes through the dorsal root ganglia and it goes into the cord. Now, from that spot in the cord, as it's going to my thalamus and to my cortex, where is it? Okay. You can read it right off the chart here. What did I call this pathway?
The anterior.
I call it the spinothalamic pathway. Did you hear that? A spinothalamic sensory pathway carrying the Pacinian corpuscle information to the brain. And I typed these in for you so you can see them. If this is crude, deep pressure, touch information, where is it? It's on the anterior side. Do y'all see it? The anterior spinothalamic tract, right along the anterior edge of the spinal cord. Now that's number one of dozens of these that you will learn. I want to introduce it to you. It gets you warmed up for the nervous system by bringing this up right now. You now know some general anatomy things about the spinal cord, don't you? What would you find, from the histology we looked at, what would you find in the ventral horn of the spinal cord? That was the neuron that you looked at on the histo test yesterday. What if I called it a lower motor neuron? Yes? Where did I get that section? Here, in the ventral horn. Now, if all the stuff here is sensory, then what is all this stuff? Motor. So later, we're going to come from the brain to here, and then go out to where? If it's a lower motor neuron, it's going out to skeletal muscle. Yeah. Okay, so you're just getting introduced to these things. We're getting you warmed up as we go. Now, here are the sensory pieces of skin. This is out of chapter 13 in your text, but it's skin information, so you need to learn them. The epidermis has free nerve endings. Does it surprise you to learn that the stratified squamous of the epidermis has innervation? No. What would it not have? Circulation, right. Okay, so this is true. These free nerve endings are true not only in the epidermis and the mucosa. Y'all know what that is already? Didn't you learn that? What is the lining of the pleura, peritoneum, and pericardium? The first layer lining. What is it? Yeah, simple squamous epithelium. Now there's connective tissue there too. Okay, that's what he means here. What's what she means by mucosa. The cornea has these things, the epidermis, glands, ligaments, tendons, free nerve endings, both epithelial and connective tissue. We also know that about these special modified things. We saw them. These Merkel discs that are found in the basal, the stratum and salvia of the epidermis. You know that there are free nerve endings, these plexus things, that are found around hair roots, in and around hair follicles. These are normally the ones that tell us you got a mosquito on your arm. Now those are free nerve ending type things, unencapsulated kinds of endings. We do have in the skin encapsulated. Now these are fascinating little pieces of anatomy. Each one of them has its own little capsule and it responds in very different ways to different things. And there are three of them. The first group are the Meissner's corpuscles. Now before you read it, can you tell me where in the skin the Meissner's corpuscles are? Where? In the dermis. Where in the dermis? Close to the top. Specifically in the, I need the anatomy word. In the dermal papillae. Yes. So if you had to guess what kind of touch they would be sensitive to, lighter or deeper, what would you say? Okay. So light pressure discriminative touch. If you learn the anatomy, you learn the physiology by default. Okay, so these are found in the dermal papillae of hairless skin, the nipples, genitalia, fingers, soles of the feet, eyelids. The Pacinian corpuscles. Now don't read it. Where are they? Okay, I need anatomy words. What layer of the dermis? The reticular layer of the dermis. Deep in the reticular layer, just before you get to the hypodermis, right? They're deep. So what kind of pressure are they going to be sensitive for? Deep pressure, yeah. So again, subcutaneous tissue of skin, periostea, I don't know what that word means, Mesentery, tendons, ligaments, all these words should look very familiar to you where these descending corpuscles. Now there's one more that the picture in the chapter didn't have, but it's important. These guys right here, these Ruffini end bulbs or Ruffini end organs, they're encapsulated receptors, and they're different than either the Meisners or the Pacinians. They do in fact detect deep stuff better, but their function, this is not a physiology class, but their function is different. We know that they are slowly adapting. So the initial information when you got touched deep is Pacinian. As you stay there, you know you stay there. Because there are slowly adapting receptors in the dermis. These guys, these Ruffini guys, that will tell you you're being touched deeply for a prolonged period of time. Look at the difference in how they're described. They are slowly or not adapting, which means they don't forget they're being touched, right? After a while, I don't know my watch is on my arm anymore, right? I forget my glasses are on my face. Those are rapidly adapting receptors. They forget. There's a physiology behind that. It's interesting. But these guys don't forget. They continue to send action potentials up the pathway as long as you're pressing on them. They're deep in the dermis, hypodermis, and in the joint capsules, the Ruffini corpuscles. All right, a few excruciatingly boring pieces of anatomy. 2 or 3 questions tops on your exam. The language of nails and hair. I dread these, frankly. I can't think of any fun stories to tell about fingernails and hair. These guys are scale-like modifications of the epidermis. Nails are on the distal and dorsal surface of the fingers and toes, and everybody knows that. Okay, so let's just label the parts of it so I can use them on a test question. There's a free edge of the nail that extends off the distal end, and that free edge is also known as the hyponychium. Hypo means under, and so it's really a reference to the place where dirt gets. Can I say it that way? The hyponychium, where the dirt gets. The nail cuticle here is known as the eponychium. It's really just an extension of the epidermis over the proximal end of the nail. It is not where the fingernail comes from. I think that's probably the biggest confusion for what you mean by cuticle or eponychium. The fingernail actually comes from the root and the nail matrix, which is underneath the cuticle. It's deep to the cuticle. And so cells are produced here that create the fingernail. I think one anatomy question that should be answered here is why does the fingernail, if it's an epidermal derivative, if it's really epidermis, in fact, a fingernail is really stratum corneum. If you peel a fingernail off, then you would find stratum granulosum, stratum lucidum, stratum spinosum, stratum basale. This is the dead keratin-filled cells of the fingernail. Why is it different in touch to this? And the answer, like everything else in anatomy and physiology, is the structure of the protein. In this case, it is keratin. It's a different structural arrangement for the protein keratin. And so this is a keratin-filled stratum corneum. The fingernail comes, it protrudes upward from the root of the nail matrix, underneath the cuticle or hyponychium. All right. I didn't mention lunula, this little half-moon shaped structure right here. Extends off of the cuticle on the proximal end. So, eponychium, hyponychium, nail matrix, keratin, and lunula. These are the anatomy words of the fingernail. All right, now hair. There are various types of hair. Vellus hair is this fine body hair found in kids and adult females. Terminal hair is coarse. But I'd like to say a little something more about the anatomy here. Because if you looked at hair, you could actually tell this. if you look at the hair microscopically and the hair is flat, then when you look at it on the person's body, the flat hair is kinky, curly. If you look at the hair and it's oval, like that, if you look at the anatomy of the hair and it's oval like that, then the hair tends to be wavy. And if you look at the hair and it's perfectly round, like that, then it's coarse. So all of mine are round. It just sticks straight up. It's hard to brush coarse hair. So the anatomy of the hair, if you look at it histologically, it can tell you a little bit about what it will look like in the gross anatomy appearance. Now, hairs have an anatomy, of course. From before, we pointed out to you that hair is an epidermal derivative. Remember that? And that's true. You can see the epidermis here. But there's also, you couldn't see it on the other picture, but you can see it on this one. There is some dense connective tissue. It's irregular, so it's part of the dermis that surrounds the hair. So it's almost as though the hair has pushed its way down from the epidermis down into the dermis. And the dermal dense regular tissue creates a sheath around it. So this outer layer of the hair, learn the layers from the outside in for me. It's a connective tissue root sheath on the outside. It's part of the dermis. The rest of the hair is actually an epidermal derivative, so it looks epithelial, right? See this epithelial look to these tissues here. And there's several layers. There's an outer layer and an inner layer called the epithelial root sheaths, and then which are part of the cortex of the hair. And in the medulla, the central region, which is arguably the most interesting, the central region of the hair here, the medulla, the cortical, the cortex, the medulla, this portion right here, is where we really can explain things like hair color and hair shape and vascularization stuff. So if you look deep in the medulla of the hair shaft, you will find that part of the dermis extends into the bottom of every hair. There's a little papillae, a little hair papillae at the bottom of every single hair, where the dermis actually sticks up inside it. Now, it doesn't show this on this picture, but that's where the blood supply is. So all the nutritional information to the hair, all the stuff that the hair cells need to survive is here in the dermal papillae. And so all the hair cells themselves, very similar to the way your skin does, come from the bottom up. And so you will find a very highly mitotic rate for the cells here around the hair papillae, or the hair bulb, or the root. This little papillae is just the extension of the dermis into the anatomy of the hair bulb or root. And you can see these cells now that are being produced here, there's lots and lots of them dense. They're going to migrate upward as the hair grows and starts to extend out through so that you can see it. Now, the last thing to point out here, you can see here around the hair papillae, where the dermis and the blood vessels are here for the hair, you can see these little cells that you will recognize. They're labeled here. Melanocytes. How would you explain hair color then? Same way you explain skin color, the presence of melanocytes. Now, there are a few other things. There's this. There's a different molecule produced by some modified looking cells here that are rare genetically in individuals with red hair. Those cells produce a molecule you all need to know. It's called trichosiderin, which is a red color of red hair. But most hair colors are created by melanin. And you can see, I want you to know where are the melanocytes. They're right on top of the dermal papillae, right in that first cell layer. Now, here's the really interesting part. I'm going to pick this up on Monday. The interesting thing to talk about in hair is how you make them stand up. And you guys know this already, right? Hairs stand up because they're attached to muscles. And these muscles are called erector pili muscles. Now, here's a histo shot up of here just to prove to you this is muscle tissue. Here's the erector pili muscle, and there's your hair follicle right there. And these erector pili muscles normally attach low on the hair and high up toward the dermal papillae. And when they contract, they pull hair follicles vertical. Now, when we start this on Monday, this is my opportunity, right? The erector pili muscles in the skin. This is my opportunity to talk to you about the different anatomies for controlling smooth muscle. Now, erector pili is smooth muscle. So, you tell me, if I'm going to control skeletal muscle in my body, everybody knows that's under voluntary control. What's the anatomy word? I'm going to control skeletal muscles. What? No, it's the other one. Somatic. Somatic is voluntary, skeletal muscle control. If I'm going to control things autonomically, what two divisions will help me explain the control of smooth muscle and cardiac muscle? I've only said them once in this class, so I'm not upset that you don't remember. They're the autonomic systems. They are beautiful, sympathetic and parasympathetic systems. All right, so I would like to talk about the differences in the way the somatic nervous system controls skeletal muscle, because we have to do that, a lot of that, and the way the autonomic nervous system controls smooth muscle at this point. I've already shown you a pathway for a Pacinian corpuscle delivering information to the brain. Now I want to come from the brain and control a muscle. And the first one on my list is the erector pili muscles of the skin. It happens to be an example of what we will call a multi-unit smooth muscle. The only ones I've introduced you to so far are the single-unit smooth muscles. And can you see already the differences in how the autonomic nervous system is going to function here? Can you see it? Okay, so we're going to talk about the details of the innervation of the erector pili muscles and extend that into the other types of muscle in the body on Monday.